Well, hello, everybody. It is our pleasure on behalf of Precisionary Instruments to welcome Dr. Joseph Ransdell, or Joey, who is our guest speaker for this webinar. So he is going to talk today on investigating how sodium channels regulate the firing of cerebellarpurkinje neurons and how you require healthy brain slices to do that. So a quick background, Dr. Ransdell completed his undergraduate degree in neuroscience at the University of Minnesota. As a true Westerner, he completed his Ph.D. at the University of Missouri-Columbia, and then he studied the regulation of membrane excitability in crabmotor neurons, so a very interesting animal model. After his graduate degree, he joined the lab of Jeanne Nerbonne at Washington University. And there he continued using electrophysiological methods to test how ion channel accessory proteins regulate the excitability of various types of mouse central neurons. And then recently in 2020, Joey started his independent lab at Miami University in Oxford, Ohio. And he continues to work with mouse models. And he and his team has focused on the regulation of voltage-gated sodium channels, and then also the contribution of these channels to the function of cerebellar circuits throughout health and disease. Specifically in today's webinar, Dr. Ransdell is going to explore how the compresstome vibrating microtome made by our company is used to produce healthy brain slices for his electrophysiology experiments. And specifically, he studies adult Purkinje neurons in mouse cerebellar brain slices. And with that, Dr. Ransdell, I want to hand it over to you. And thank you so much for being here.
And can everyone hear me okay?
Yes.
And I will say this, I'm wearing a headset with the mic, but I've had issues with it going out. If it does, please interrupt me and I can switch to a different mic pretty quick.
Great.
So thanks a lot for having me. And as you and I were just discussing, I mean, I'm happy that there is some turnout here because of course SFN and beautiful San Diego is going on right now. So I'm grateful for anyone that's able to attend now. And then of course, I hope people are able to view the webinar recording at a later point in time. Let me go ahead and share my screen. So I shared, okay. And so can everyone see my PowerPoint?
Yes.
Okay, thank you. Okay, and so, yeah, so the title of my talk, and I was reading it after I had sent it to you, and it's kind of a mouthful and doesn't really roll off the tongue, so I apologize for that. But it's investigating how sodium channels regulate the firing of cerebellarpurkinje neurons, and then I kind of tagged on requires healthy brain slices. And that last portion is what I'll really be focusing in on today. So this is the first time I've ever given one of these webinars kind of sponsored by a company, but I've attended several. And I know that personally, what I get the most out of is hearing from the personal experiences of other scientists and kind of figuring out what works for them so that I can kind of utilize that information in my own studies. And so I kind of formulated this talk with that in mind to kind of present to you kind of what we as a lab went through in kind of organizing our equipment in order to successfully produce these studies where we're looking at the electrical activity of these mouse Purkinje neurons. And so I thought how I would do it is kind of introduce my lab and then go through our experiences with the compressstome, and then towards the end, kind of provide you with some experimental result information. And of course, you know, I'm an open book. I'm happy to discuss more of those experiments or our results or anything. So people can feel free to email me after, or maybe we'll have enough time for a discussion afterwards. So a little bit about our background, right? So we're at Miami University. And so more specifically, I work in the biology department here at Miami. And then within the biology department, I'm in the Center for Neuroscience and Behavior, or within the group that focuses on neuroscience and behavior. And I threw this image up of Miami University, and I chose this image specifically because it's an image of Miami during the fall. And I think that kind of highlights the fact that we're Miami University in Oxford, Ohio, right? So we're not in Miami, Florida, because that's kind of a common misconception. Okay, so as you mentioned, I started this lab in 2020, and kind of what, if we kind of had to break down the lab and what the lab goes after into one central question, it's kind of this basic biology question. So we're interested in how the electrical activity, right? So how the membrane excitability of a given type of neuron is going to be produced and maintained. And then I kind of tag on after that during health and disease. And that's kind of reflective of the fact that we use mouse models specifically because we can manipulate ion channel expression in those models to kind of mimic neurological disease. So using those mouse models, we can then study, okay, in this model of neurological disease, what happens to the electrical activity of neurons, and then how is that reflected in animal behavior? So if you're not familiar with the study of membrane excitability, I kind of threw this in to just show the extreme level of variability or the heterogeneity that we can see in the electrical output of neurons. So for instance, this is a thalamocortical neuron. You can see it's firing a pretty fast burst of action potentials. And that's very different than the electrical activity of this medium spiny neuron, which would be in the striatum, which has this kind of delayed firing with a depolarizing current injection. And then for good measure, I also threw in a non-neuronal cell type. So this is a human ventricular myocyte, and it's generating its own action potential waveform. And then finally, I wanted to put up an example of the electrical activity of cerebellar Purkinje neurons. And so this is what I'll be focusing on during today's talk. And so if you just look at the timestamp here of 2 milliseconds and then compare that to the cell types that I showed before. So with the myocyte, we have 100 millisecond, 200 millisecond timestamp here, and a 50 millisecond timestamp here. You can see one of the kind of unique properties about cerebellar Purkinje neurons is that they have this capacity to fire action potentials repetitively at very high frequency. So anywhere from 50 to 100 hertz without any sort of depolarizing current injection. Now, the other kind of cool or unique aspect of cerebellar Purkinje neuron excitability that I want to point out to you is that this capacity to fire these repetitive action potentials is not dependent on any sort of synaptic drive. So, for instance, you can see in this micrograph here of a Purkinje neuron, if we eliminated all of the synaptic input to that cerebellar Purkinje neuron, it would still maintain its capacity to fire these repetitive action potentials spontaneously. So it's kind of an interesting and unique property. One, that they can fire very quickly, and two, they can do so spontaneously. So they have sort of pacemaker depolarizations driving this repetitive firing. So it makes for a pretty interesting cell type if we're trying to figure out, okay, how is this electrical activity generated? But it's also a pretty important cell type, and that's because of the way it exists in cerebellar circuits. So this green highlight here is just denoting the cerebellum. And so if we slice the cerebellum both coronally and sagittally in this kind of cartoon depiction, you can see the Purkinje neuron layer shown here with these big green cells. And so Purkinje neurons are unique because all of the input or all of the information entering into the cerebellar cortex is actually integrated by the dendrites of these cerebellar Purkinje neurons. And in addition to that, the Purkinje neurons have their axons that then leave the cerebellar cortex and innervate deep cerebellar nuclei. But these axons are the sole outputs of the cerebellar cortex. And so that's kind of another interesting thing and a reason that we like to focus in on this cell type. So if you consider, right, that these Purkinje neurons are integrating all of the information coming into the cerebellar cortex, and then they're the sole output of the cerebellar cortex. You can imagine that if that electrical activity, right, if these repetitive action potentials that the Purkinje neurons are generating, you can imagine that if this electrical activity were disturbed in some way, right, so if there was a mutation maybe in an ion channel that caused deficits in this electrical activity, it wouldn't just be a deficit in this electrical activity, right? It would actually screw up the cerebellar circuit altogether. And so what makes these Purkinje neurons a nice cell to focus in on is because if we have an animal model that has deficits in Purkinje neuron firing, we can actually see those deficits kind of reflected in the mouse behaviors that are associated with cerebellar functioning. Okay, so I kind of started with our main focus, right? We're interested in how this repetitive electrical activity is generated in cerebellar Purkinje neurons. And we're interested in that, one, because it's just a cool thing to kind of think about, but two, because Purkinje neurons play this central role in the cerebellar circuitry. So to kind of get at what underlies this spontaneous electrical activity, we look at the suite or the repertoire of expressed ion channels that are inserted into the Purkinje neuron membrane. And so, for instance, in mammalian neurons, there's over 100 types of different ion channels that can be expressed and inserted into the membrane. So we're interested in what combination of these ion channels can generate this type of electrical activity. But then to make matters more complex, right? It's not just the various pore-forming ion channel subunits that are expressed and inserted in the membrane. We also know that these ion channels function as macromolecular complexes. So various accessory proteins can bind and regulate the functioning of any given ion channel. So for this particular talk, we're focusing in on the voltage-gated sodium channels. And it seems to be a nice thing to kind of study because there's a lot that we still don't understand. So of course, we know that voltage-gated sodium channels are really critical for electrical activity because we know that those voltage-gated sodium channels are responsible for this rapid upstroke of the neuronal action potential. But then what we'll kind of jump into later in this talk is that we know these voltage-gated sodium channels, at least in Purkinje neurons, are responsible for tuning this high rate of repetitive firing. And so these last two images that I've kind of put up, this would just be a cartoon depiction of the pore-forming alpha subunit of a voltage-gated sodium channel. And then if we kind of slice that open and looked at it, looked at the cross-section, we can see the various transmembrane segments of that pore-forming alpha subunit. But we can also see that there's various accessory proteins that we know bind and regulate the functioning of this sodium channel. So that's kind of the second part of what my lab's kind of jumping into, is trying to understand how each one of these accessory subunits regulates the functioning or the gating processes of this voltage-gated sodium channel, and then how that in turn affects the electrical activity of these cerebellar Purkinje neurons. And I mean, I think we have a small enough group. If there's questions, I certainly don't mind if someone wants to stop me and we can discuss something further.
Definitely, we can do kind of like a small round table. So
yeah, absolutely. Okay, so our focus are these Purkinje neurons in the cerebellum, and as we've discussed, we utilize these mouse models to do that. And so in order to kind of get at the electrical activity of these cerebellar Purkinje neurons, we generate or produce these sagittal brain sections from the mouse. So we quickly decapitate a mouse, quickly slice sagittal brain sections, all while the sections are in oxygenated artificial cerebral spinal fluid. So we're keeping the cells nice and happy within those brain sections. And then of course, personnel in the lab can take these living sagittal brain sections and put them on an electrophysiology rig and then generate electrical recordings from the cerebellar Purkinje neurons. So for instance, this is Sam Brown, which he'll come up later on in this talk, but he was the first graduate student to join our lab group. And he's recording from a cerebellar Purkinje neuron here. And if you look closely, you can see on his screen, he's injected some depolarizing current into that Purkinje neuron. to evoke sustained firing from the Purkinje neuron. And then this is just kind of a cartoon depiction of what he would be doing, right? He would be moving this glass electrode and actually patching on to the living Purkinje neuron to record its repetitive action potential firing. Okay, so the final point, and I hope that you're all still with me in terms of, you know, how we go about doing this. But the final point that I wanted to make in discussing this is a feature of Purkinje neurons. And that's that Purkinje neurons are kind of notorious for being difficult cells to keep alive. And so that means a couple of things for us. We know that they can be easily damaged during slicing. So, you know, trainees in the lab have to learn quite a bit on methods in order to appropriately generate these sagittal cerebellar sections. And also they have to be pretty quick into generating these cerebellar sections, because we know that the Purkinje neurons will start to die off fairly quickly if it's not done correctly. And then in addition to that, Purkinje neurons are difficult to record from because they seem to have this tough extracellular matrix atop them that needs to be cleared away before that patch recording electrode can approach the cell membrane. And so, for instance, I've recorded from cortical pyramidal neurons and hippocampal pyramidal neurons. Whenever I'm clearing off debris from those neurons, I might use 0.1 milliliters of positive pressure to kind of blow away any debris sitting atop the cell. In Purkinje neurons, we routinely use 2.5 milliliters to 3 milliliters of positive pressure. So a huge increase in positive pressure that's necessary in order to clear away that debris that sits atop the Purkinje neurons. What this also means is because this effect of the extracellular matrix kind of gets worse as the animal ages, we're really limited to only recording Purkinje neurons from these cerebellar slices to about 8 weeks of age. If the animals are older than that, we run into such severe difficulty, right, getting to the Purkinje neurons that it's not really worth pursuing. Okay, so again, we're interested in patching these Purkinje neurons, but they're kind of notorious for being difficult cells to generate these patch recordings from. And so we've already talked about that I started my lab at Miami in 2020, and I don't know if how many new PIs or maybe postdocs that are finishing up their position and entering the job market, but you know, this is something that really hits you hard, right? When you're starting a new laboratory, right? You arrive, and this was kind of a special circumstance, given that we were in the middle of the COVID epidemic at the same time. But arriving at Miami University, this was our shared lab bay. This is what it looked like, right? Completely empty. And then this is this image to the right. This is our electrophysiology room. And so you can imagine you're kind of geared up, you know, the experiments that you want to do, but when you arrive, this is what you're kind of faced with. And so during this next part of the talk, I'll kind of be taking you through how we put together the lab to ultimately end up with this. Now in 2022, you can see that our molecular lab bays are pretty well stocked. And then in our electrophysiology room, we now have 3 functioning rigs. And then if you look closely, and I think you can see my mouse cursor, I'm kind of circling it. You can see our compresstome right here, where we generate those sagittal cerebellar sections to then quickly move to the electrophysiology rig. Okay, so before getting into the kind of equipment purchases, I'll take you through kind of what we were driving forward to, right? So I knew that I wanted to study kind of how this electrical activity was generated, but I didn't know the specific experiments, right? So if we rewind to before 2020, I was a postdoc at Washington University in St. Louis. And at this point in time, I was working with Jonathan Marino and John Silva, who are investigators in the biomedical engineering departments, and then also working with my advisor, Jeanne Nerbonne, in cardiology. And so we were working together to generate a mathematical model that could accurately reproduce the properties of voltage-gated sodium currents in these cerebellarpurkinje neurons. And so to do that, we generated this Markov kinetic state model. And this was recently published. I forgot to put the reference up here, but this was recently published in 2022 in the journal eLife, if you're interested in looking at that. But we published, or we had produced this Markov kinetic state model, which was a nice mathematical representation of how these voltage-gated sodium currents operate in these Purkinje neurons. So I kind of walked into Miami University with this model in hand. And the model does a pretty good job. So this is a voltage clamp recording from a cerebellarpurkinje neuron. And so what you can see is with that initial depolarizing step to 0 millivolt, we're activating a fast transient or a fast inactivating sodium current. And then on this repolarization step, we're evoking the resurgent component of the voltage-gated sodium current. So you can see that resurgent component turning on. And then over time, that resurgent component decays, and we're left with just a persistent sodium current or a non-inactivating sodium current. And so again, this is a experimental recording of a voltage clamp protocol from a mouse cerebellarpurkinje neuron. And then this is the performance of the Markov model that we developed. And as you can see, it does a pretty good job of reproducing the sodium current properties. But kind of going back to our original question, we're interested in how these sodium current properties actually contribute to the firing of a Purkinje neuron. And that's kind of an easier, it's an easy thing to think about, but it's a hard thing to kind of show, right? So we have this voltage clamp recording of the sodium current, and we can measure its kinetic properties and its voltage dependence and things like that. But then we're kind of left with okay, well, if there's channels that are producing this current in the Purkinje neuron membrane, how are they actually contributing to these firing properties? And to really get at this question, we can use another electrophysiological technique known as dynamic clamp. And this is kind of what I wanted to jump into right away with starting our lab at Miami University. So in dynamic clamp, we start with our mathematical model, right, that represents the current that we're interested in. And then if we have a patch recording of the membrane potential of a given neuron, right? So here we're patched onto a Purkinje neuron, and that Purkinje neuron's firing action potential is rapidly. What we can do is feed that voltage signal into the computer, right, that has our modeled ionic current, right? And so with that voltage signal fed into the mathematical model, the model can calculate or compute what the current would be doing at a given membrane potential or a given voltage. And then that can be interfaced with our current injecting electrode to actually add that current or add that ionic conductance to a real neuron, right? So it's kind of I like to say that it's the ultimate brain machine interface, right? We have this mathematical model that's actually being added to a real firing neuron. And so you can imagine adding sodium current to this cerebellarpurkinje neuron through this current injecting electrode would probably drive up or scale up its repetitive firing rates. So before we could do any of that though, right, we're stuck with this empty lab. So we have to get the equipment that's appropriate to carry out these dynamic clamp studies. So I was lucky enough in 2020 to have a graduate trainee join the lab group right away. So when I arrived, this graduate student named Sam Brown arrived, and I kind of purposely added his picture on top of the empty lab space, because now not only is there an empty lab space staring at me, but I also have Sam Brown staring at me, right, waiting on what he's going to be able to do. So right away, we started buying or purchasing equipment to begin these experiments. And so in these equipment purchases, I decided to take two kind of big risks. The first was I purchased a Sutter patch clamp amplifier. And I say that this was a risk because during my previous experiences as an electrophysiologist, I had always used molecular devices amplifiers. And so I was used to those amplifiers, and I was used to their software. But I decided to go with the Sutter patch clamp amplifier for a couple of reasons. One, it was less expensive than our molecular devices amplifiers. But also Sutter, this is a fairly newly developed amplifier. They actually built in dynamic clamp hardware as well as built dynamic clamp protocols into their software. So when I purchased this amplifier, it kind of set me up very nicely to carry out these dynamic clamp experiments. Now, the other kind of risky behavior was I purchased this Precisionary compresstome. And just like with the Sutter patch clamp amplifier, I looked at it as kind of a risk because previously as a postdoc, I did not use Precisionary compresstomes. And, you know, if the compressed-tome didn't perform appropriately, right, I would be stuck getting these cerebellar tissue slices that had dead Purkinje neurons. And so I looked at that as kind of a risk as well. So, I mean, the safe bet, right, if I wasn't willing to take that risk, would have been to purchase a Leica vibrotome. So this VT1000S Leica vibrotome, that's what I used as a postdoc. And I can say, you know, it worked. I generated sagittal cerebellar sections and the Purkinje neurons were alive. But I also often ran into problems with this vibrotome. And so I wasn't completely happy with it. And so while I was kind of looking to purchase equipment, I started looking around and seeing if there were any discussions out there about, you know, which vibrotome is the best. And so for that, one of the sources was ResearchGate. And I don't know if you're familiar with ResearchGate. It's kind of this social network for researchers, but they have question forums, right? And I found some people that had asked this question before, like, what's your favorite vibrotome for generating, you know, brain slices that are healthy? And so I was reading answers in this question forum, and I stumbled on an answer given by Jonathan Ting. And so interestingly, I think Jonathan Ting has previously presented a webinar for Precisionary.
He did. Yeah. He's been with us for almost 2 decades. And his webinar topic was, you know, a decade of reflection on brain slicing.
Right. Well, and so, you know, I'm looking at these answers and you want to be able to have some confidence in the answers that you're reading. And so when I saw Jonathan's answer, it was really well written and it looked like he had given it a lot of thought. But also I had known Jonathan, I don't know Jonathan Ting personally, but I knew who he was because I had read his manuscript. I guess it's not really a manuscript, but it's in the Journal of Visualized Experiments, where he basically discussed a new way and what he, you know, said was a better way for generating healthy brain slices. And so when I saw Jonathan Ting reply, I knew that this is someone that had given a lot of thought to what actually produces these healthy brain sections. And so I won't go through his, you know, it was a pretty long answer, but I won't go through all of the details. But he basically said the Leica 1000S isn't great, the Leica 1200 is better, but he basically said that the Precisionary VF200 seems to be just as good, if not better. And also that the VF200 is very easy to use and very easy to acquire parts for if anything ever breaks. And so, you know, that made me happy, and it made me actually start exploring the Precisionary vibrotomes. And what I quickly realized, right, was that the VF200, I think, had been replaced at that time by the VF3100Z is the model number.
That's right. Now, the VF200 is a much older model. It's one of our first ones. And so we've had newer models come out.
Yeah, that's what I kind of figured was going on, right? That they had just updated the model. Now, I wasn't completely sold at this point because remember, Purkinje neurons are kind of a special cell type. And I didn't see any information on Jonathan actually patching Purkinje neurons. And I knew that they were kind of notorious in terms of maintaining their health. And so I did a little bit more digging, right? And I eventually found a dissertation from a student in Morten Rostad's lab. And I don't know if I'm pronouncing that right, but he's an investigator at Emory University. And then this took me to publications from Morten's group as well, where they had used the Precisionary compressed tome and actually generated recordings from mouse or I think it was actually rat cerebellarpurkinje neurons. And so I emailed Morton, and he was kind enough to email me back to give me his experiences. And he had also used the Leica vibrotome as well as the Precisionary. And basically, again, I won't go through the whole email, but his comments were that he had used both, and the Precisionary compresstome seemed to generate better cerebellar slices and better Purkinje neurons for recordings. And so that's those two pieces of information together kind of sold me, right? That I would skip going with the Leica 1200S model and go with the VF3100Z. Now I'll also mention, right, as part of our purchasing department, right, we're required to get quotes of multiple devices. And so I did get a quote from the Leica, and it was kind of an added benefit that this VF3100Z ended up being about half the price as the Leica Vibrotome. So we got the VF3100Z, and Rachel Falconer, I don't know if she's still with Precisionary, but she was our rep, and she was very helpful in training the graduate students in the lab and how to use it. And it seemed fairly easy to use. The one thing that I've noted right away is that it's much faster in generating these cerebellar tissue slices than my experience with the Leica. And so I can only imagine that probably also improves Purkinje neuron health for later electrophysiology experiments. Now, a few months in, we had issues with one of the parts on this VF310, and we contacted Rachel, and I think we were still under warranty, but during that time, she basically offered to swap out their newer model, which was the VF500Z, so that we could directly compare the performance between the two models. And so they sent us this VF500Z, and we directly compared the two models. Now, I'm sure I'm not going to be going through all of the differences that the engineers are aware of, but what was clear to us right away is that the VF500Z has an adjustable blade angle, whereas the VF3100Z had a fixed blade angle. Now, I should mention, with the VF3100Z, we were getting healthy cerebellar sections, but what we found was that our sections were more uniform, and our cerebellar Purkinje neuron health was better with the VF500Z. Now, the differences between the two weren't like huge for us, right? We were both were giving sections that had Purkinje neurons that were healthy and good for electrical recordings, but the slices just tended to look better from the VF500Z. And so then we basically held on to this model. So this is the model that we're currently using. And I was actually just on Precisionary's website, and I think this has been updated again to now it's the 510Z. But anyway, this is the model that we're using right now, and we're happy with. So we have our Sutter patch clamp amplifiers, and we have our Precisionary compresstome so that we're able to get these cerebellar sections. And so now we're back to those dynamic clamp experiments. And so if you remember exiting the postdoc, right, I had this model in hand, and I was able to hand that model off to graduate student Sam Brown. And then he was able to kind of, once we had assembled the electrophysiology equipment, he was able to start generating these cerebellar sections and start applying this model using the Sutter patch dynamic clamp software. So again, this is the modeled sodium current. And so Sam is basically adding this sodium conductance to a cerebellar Purkinje neuron in current clamp mode, right? So we're looking at how this added conductance is then going to impact the firing or the electrical activity of this cerebellar Purkinje neuron. And so that, just adding it is one thing, right? And what you'll see in a second is when we add that sodium conductance, which is a depolarizing conductance, it increases the firing rate of those cerebellar Purkinje neurons. And that's not really a surprise to anyone. So we wanted to actually ask some biological questions. And so the first question that Sam asked was, how does changing the magnitude or the proportion of this resurgent component of the sodium current affect how much it influences firing frequency? And so he developed another model where all of the properties of the model were similar, except that resurgent current is now smaller. And then what you can also see is that in this updated model shown in green, his persistent current is also smaller. So he has a reduced resurgent sodium current and a reduced persistent sodium current compared to the original model. And then he developed a third model where that resurgent current is much larger, right, than the original model. And so what his first kind of experimental question was how does scaling the amplitude of this resurgent component impact the firing rate of these cerebellar Purkinje neurons? And so the results were actually pretty surprising. So if you look at the black dots, and I'm sorry, I should have, oh, they are labeled. So the black dots would be no model. So this would just be the spontaneous repetitive firing of an adult Purkinje neuron in a cerebellar section, right? And you can see it, they're firing quickly, like they're known to do, between 50 and like 130 hertz or 130 times per second. And then he added the normal model, which in this case we're just referring to as the intrinsic gating model, right? And so when he adds that normal model, it increases the firing rate of those Purkinje neurons, right? They go up. And again, that wasn't a huge surprise to us, right? We're adding these depolarizing sodium conductants. It's probably going to increase the firing rate. But then when he switches to the green model, so this green model, the transient component's the same, but it has a reduced resurgent component, and it also has a reduced persistent component. When he adds that green model, what he sees is that there's a significant drop in the firing rate from that original model, referred here as the intrinsic gating model. So that kind of, again, was not surprising to us, right? We're reducing this resurgent component and the persistent component. That makes sense that it would result in a drop in repetitive firing. And then in those same cells, right, he's adding the larger resurgent component model. So now in this model, the sodium current has an amplified resurgent component, and its persistent component's right about the same as the original model. And this is where things kind of became interesting, because we didn't see a significant change with this larger resurgent sodium current model. And that was kind of unexpected because, and I haven't given a whole lot of background on this because I wanted to focus on kind of the tools side of things, but at least the common thought in terms of this resurgent sodium current is because it's a depolarizing current that turns on during the repolarization of the membrane, it's thought that more resurgent current should scale the repetitive firing rate of these neurons. And here we're not seeing that. We're, when we have this something like, I think it was like a 300% increase in the resurgent component, we don't see a dramatic shift in the firing frequency of these cells from the original firing frequency. And so then we kind of, asked the question, well, why doesn't this larger resurgent current drive these higher firing rates? And we came up with the hypothesis that maybe it's not the magnitude of this resurgent current component. Maybe it's the magnitude of the persistent current. Maybe that's the ultimate driver. Because if you recall that green model, right, the model shown in green here, it has a reduced resurgent current, but it also has a reduced persistent current. So we thought, well, maybe it's that reduction in the persistent current that's causing this model to drive lower rates of firing. So to test that, and so I'm just adding that in text here, right? Maybe it's the persistent current after the decay of the resurgent current that's the important parameter. So to test that, Sam made an additional model. So in black, we're showing the original sodium current model. And then in blue, we're showing this last model that he made where he actually removes most of the persistent component from the current trace, although he maintains most of the resurgent component. right? So now he's directly testing what's the difference when we just remove that persistent current. And what we saw is that compared to the original model, that removing that persistent current results in a significantly lower firing rate. And I'm showing an N of 4 here, which Sam probably hates, right? Because he's expanded these studies and he probably has 30, 40 cells now that show the same effect, that when we reduce this persistent sodium current, we can actually scale down the repetitive firing rate in these cerebellar Purkinje neurons. Okay, and with the conclusions slide, sorry, I went a little bit faster than I had hoped for, but I think that's okay because I think we could, maybe have a discussion afterwards or something. But in terms of the tools side of things, what I wanted to point out, right, is that this VF310-0Z and the VF500-0Z are sufficient, right, for generating sagittal cerebellar sections that have healthy mouse cerebellar Purkinje neurons. And then kind of to the biological question side of things, right, what we found using these dynamic clamp studies was that this peak resurgent sodium current doesn't seem to be a critical parameter in tuning the repetitive firing rate of these cerebellar Purkinje neurons. But instead, it really seems that the critical parameter is the proportion or the magnitude of this persistent sodium current. Okay, for acknowledgments, and I kind of mentioned these people throughout, but Jonathan Marino, Jeanne Nerbonne, and John Silva were the people that I worked with at Washington University to kind of develop that initial sodium current model. And then, of course, Sam Brown was the first grad student to join my lab group at Miami, and he's since taken that original sodium current model and produced additional models, right? I think I presented 3 of those models in this talk, but he's produced even more than that, right? Where he's basically manipulating various sodium current properties to see how it affects Purkinje neuron firing in dynamic clamp. And from Precisionary Instruments, I would think Rachel Falconer, who was our initial sales rep, and I think I mentioned she helped us through the training and she helped us acquire the BF500 model. And then Abby Chu for arranging for this webinar. And then, of course, our funding, which includes my startup funds and NIH R15 funds. And I threw my email on here, and I hope, if anything struck an interest or you want to discuss anything that you don't want to discuss right now, I hope you'll send me a message over email. And with that, I'd like to thank everyone for attending.
Thank you so much, Joey. I do want to open up the floor and see if anybody had any questions, comments, anything you wanted to share at all. Feel free to unmute yourself. Yes.
Yeah. Hey, mate. Thanks very much for the talk, man. Yeah, it's super eerie, actually, to be honest. Because sorry, my name's Dave as well. I just got the university code. So I'm Dave Lyons. Just started my own laboratory in the University of Manchester in the UK in May, and I think my starting experience may be more traumatic because the lab space we had was, oh my god, it was like a bomb site. So it wasn't quite as pristine as yours, which was great. But the points here, sort of the trajectory you've had has been almost eerie. So I've done exactly the same thing. Spent years with actual instruments, and then did exactly the same. Bought a D clamp the other month, and it's just arrived. I'm all excited about that. Mm-hmm. And equally, we haven't been able to buy it yet. We've gone in for the first grant, the compresstome as well. So I suppose I've got 2 questions. One about the compresstome, because obviously we very timely put this on for everyone. And the second would be about the ease with which you've manipulated persistent sodium current with the sock patch, actually. So the first one with the compresstome, if I understand correctly, it's agar embedded, is that right?
Yes.
Right. How do you find going from just simple, maybe a transcardial perfusion, blocking, and then a Leica dissection, or maybe a Camden instrument one, and the agar embedding? We kind of used to do that sort of stuff in the good or bad old days, but it did slow things down appreciably. So is it something doesn't slow the process down. You were saying it was actually more rapid, but I presume that was the actual physical topic.
Yeah, that's a good question, and it was something that Sam and I were really worried about initially, because we perfuse our animals, we do a transcardial perfusion, and then we quickly get the cerebellum out. And again, with the Leica, I'd rapidly glue it to the base and get it into cutting solution, ice cold cutting solution, right? With the agarose, right, you have to have warmed agarose, and then you have to apply that agarose before you're able to get it into that cutting solution. And so I was really concerned, one, with the warmed agarose, that would cause the Purkinje neurons to die, and two, that it would take too long getting it set in the agarose before I could get it into the ice cold cutting solution. So basically, the warmed agarose, we haven't had an issue with that killing off our Purkinje neurons. We have had to kind of become efficient in putting the agarose on and then quickly getting that inserted into the slice chamber. And that's just a matter of kind of, I know Sam has like a water bath that keeps his agarose at kind of the perfect temperature, and then he can, quickly apply it and get it put in. So I think it's just a matter of getting your equipment set up so that you can move as quickly as possible. But again, with all of that said, the warmed agarose, after slicing, the Purkinje neurons are happy. So it doesn't seem to be, a problem.
So the proof's in the pudding, that's kind of cool. I mean, it was also with Jonathan Ting stuff, so I've been using his solutions for about 6 years now, and he also just said temperature, who cares? So one of the things he was doing was, 20 degrees or whatever. So I suppose, yeah, great. That's really good to hear, actually.
Yeah.
I'm interested in. So I suppose, it's really eerie to me, actually, because literally bought the same amp and interested for the same purpose. So interested in persistent sodium current, but not with regards to intrinsic excitability, but rather its role in a sub-threshold oscillation. So I was wondering, how easy did you find to because I'm dynamic clamp naive. So
I see.
Yeah, how easy?
If you have the Sutter amplifier, it's a pretty simple process. I did dynamic clamp as a postdoc, and I did it with RTXI software. And with that, you're basically linking up two distinct computers with two distinct digidatas. I don't know how many BNC cables, and it's really difficult to work with. With the Sutter patch dynamic clamp system, it's very plug and play, right? You can set things up pretty easily, especially if you're working with Markov models. That's another thing that I was surprised that they kind of had the software set up to add Markov models. You didn't have to code them up separately or anything like that. It's a pretty new software system. And so that would be the only kind of hiccup that we've encountered, is that it's a new enough software system where they're still kind of working out bugs within it. But one thing that's kind of nice about that is that they're very receptive to any sort of problems you're having. So I don't know how many times we've contacted them saying, like, we want to be able to do this. This doesn't seem to be working, right? And so they'll basically give us workarounds if there's any bugs. And then you can tell they're trying to release updates pretty regularly to improve the software suite.
Yeah, my experience on this has been ace as well. But I know what you mean about the software. It reminds me of like Patchmaster. So it's like eye bleeding, but I was like, oh, it can be quite slowly getting there. So
it's from my work, and this is a tough thing because I don't know how much of it is that I'm just used to ClampX and I know ClampX backwards and forwards, but ClampX seems to be much more bug-free and intuitive. But again, that could just be my own bias because I worked in ClampX since I was a graduate student. So
me too, and I think intuitive is the word. So I spoke to Telly, and they were very kindly gave me sort of a guided tour of the ego, and I'm slowly getting there. But to start from scratch, which for complex I always found quite easy.
Right. Yeah, no, I totally agree.
Are there any sort of online resources you would recommend for looking at dynamic clamp, for using dynamic clamp at all?
No, I haven't run into any. No, sorry. Outside of manuscripts and just consulting people like Telly, right, on kind of a regular basis. RTXI is no longer supported because they lost their funding. And the people that used to work in support for RTXI, they used to be very helpful. But I think since they lost funding to actually administer that support, they kind of don't like to talk about it anymore. So anyway.
Okay, well, wait, thanks very much, and really appreciate your talk.
Yeah, and feel free to email me if you think of anything else.
Great. We have just a couple of more questions. It's getting busier here. One question for you, Joey, is have you ever tried maintaining the slices for long-term culture? And if so, do you have any advice for maintaining cell viability over time? And I guess a general question that I would like to ask off of that is, if you don't do culture, if you just do daily ephys, how long do your slices typically last for?
Yeah, so I don't do culture. And I'm sorry, I can't provide more information about that. I early in my postdoc, I ran into basically issues where people had found things in cultured, what were those cell types? It was hippocampal pyramidal neurons. They found certain effects of this one accessory protein named FGF14. They found certain effects in culture, right? And then when we did the same experiments in acute brain slices, right, we saw completely opposite effects. And so that really, it destroyed my my drive to work with cultured cells, because I think I would always have this worry that maybe what I'm looking at is not real.
Right.
And so, but to answer your second question, right? So we keep our brains, we section our brain slices, and we keep them at room temperature and oxygenated artificial cerebral spinal fluid, and we can keep them. I mean, I'm sure you could probably keep them for 10 hours and be fine. Mm-hmm. It usually goes past the workday for me, where I generate on average 6 cerebellar slices that I want to use. I do 350 micron slices. And once you put the slice on the rig, we heat them up to physiological temperature. And when you heat them up to physiological temperatures, you usually have about an hour before you start seeing the Purkinje neurons die off. And so, you're putting each slice on the rig for an hour. That means you have about 6 hours worth of electrophysiology ahead of you at max.
Right.
But that said, on that 6th cerebellar slice, I've encountered, tons of healthy Purkinje neurons. So that's why I say probably you could hold those slices in that chamber for 10, 12 hours before probably bacteria becomes an issue, and you probably wouldn't have any problems.
Got it. Very nice. And we actually have a question from Oscar. I don't know if you'd be comfortable unmuting yourself and asking.
Maybe no mic.
Was it not? Yeah, it's in the chat box. So his question is, I would like to know if you let the slices sometime after you, like what you do to the slices after you cut them and before you put them in ACSF.
Yeah. And sorry I didn't go into too much detail there. I didn't want to, bore anybody, but this is how we do it. We use ice cold cutting solution to actually slice, and then immediately from the ice cold cutting solution, they're moved into room temperature artificial cerebral spinal fluid, again, that's oxygenated. And at minimum, we let them rest at that room temperature slice chamber at minimum for, I'm sorry, I'm forgetting a step. You can tell I'm I'm not as involved in these experiments. Sam's probably pulling his hair out right now. Okay, so we slice them, right, and we move them to that artificial cerebral spinal fluid. At that point, we incubate them in a hot water bath at, I think, 33 degrees Celsius for 25 minutes, right? So they're in this warmed water bath for 25 minutes. And the idea behind that is that helps to loosen all of that extracellular matrix junk that kind of sits atop those Purkinje neurons. From the water bath, we move them back to room temperature, and we let them rest at room temperature for 35 minutes minimum before starting electrophysiological recordings. Sorry, I completely forgot the 25-minute water bath stuff.
Very nice. Thank you. I think that's it. So I think I'll wrap up our webinar. Dr. Ransdell, again, thank you so much. It's been a pleasure having you. Please reach out to us anytime you need something. Same goes for all of our attendees here. And we'll have this available online for all of our scientists and researchers. Good luck with your new lab, and I hope you'll fill it up really soon.
Right. Thanks a lot, everyone.
Bye-bye.
Bye.